Onset and span of cortical neurogenesis. The mammalian cerebral cortex is a laminated structure composed of a bewildering diversity of neurons arranged in distinct cytoarchitectonic fields. It is well established, but nevertheless always fascinating, that none of these neurons are generated in the cortex itself. Classical studies of neurogenesis based on the distribution of mitotic figures and development of migrating neurons in the cerebral wall of the human fetus suggests that all cortical neurons in human are likely to be generated mostly before birth. For example, Poliakonov, 1959-1965, reviewed in Sidman and Rakesh, 1973-1982, Rakesh, 2002, by Strawn et al., 2008. However, the precise data on the onset and termination of corticogenesis could not be established with the classical histological techniques alone. For example, Connell, 1939. A limitation that was overlooked with the introduction of the trishaded thymidine and BRDU incorporation as markers of DNA synthesis to label dividing cells. Brewing, Arleno, Mackless, Rakesh, 2007. Tritiated thymidine. These new methods allowed a systematic study of neurogenesis, and it was established that the neocortex receives the first neurons at approximately E10 in mice and approximately E33 in macaque and human, reviewed in Bystrom et al., 2008. These studies also showed that there does not exist a pattern between species for the timing of neuronal genesis, and while in mice, neurons are generated in the second half of gestation between E10 and the end of gestation, E18.5, in the macaque, monkey, and human, neurons are produced much earlier, mostly during the middle of gestation in monkeys and during the first half in humans, between E33 and E100 in monkeys and E33 and E120 in humans. Rakesh, 1974, 1988, 2002, Rakesh and Sidman, 1968, Sidman and Rakesh, 1973, 1982. This early neocortical genesis stands in contrast to that of the cerebellum, olfactory bulb, and hippocampus, which in the mouse terminate their development postnatally, and in primates, including human, continue to add neurons well after birth. Rakesh, 1973, Rakesh and Novakovsky, 1981, Kornak and Rakesh, 1999, 2001. In spite of comprehensive search in the adult monkey neocortex, no additional neurons are added during the animal's 30-year lifespan. Rakesh, 1985, Kornak and Rakesh, 2001, Koketsu, Mikami, Miyamoto, Hisatsun, 2003. The claim of new neurons arriving to the prefrontal, parietal, and temporal association cortices in adult macaques could not be confirmed. Reviewed in Bruning, Aureliano, 2007, Rakesh, 2002, 2006. And additional techniques, such as the analysis of incorporation of C14, has provided further evidence that all neurons of the human neocortex are generated before birth. Bardway et al., 2006, indicating that for the highest cognitive functions we use and depend on the same set of neurons throughout our entire lifespan. Rakesh, 1985, 2006.